Okay. Welcome everyone to Introduction to Statistical Genetics. Yeah, I'll be your primary instructor, Daniel Adkins. I won't bother introducing myself. I think Matt did a good job there. But I will introduce my colleague, longtime colleague, collaborator, and co-teacher of this course, which is Andre A. Shavelin. So there's Andre. Andre, is integrally involved in this course. So he basically the kind of division of labor that Andre and I have during the next 4 days is, I'm going to primarily be delivering the content, although, I'm sure he will chime in when I overlook things or he has contributions to make. But he'll kind of be in the background, and he can answer questions, any questions that arise during the course of delivering the content. And Andre is just going to be kind of triaging those questions as we go through the course. And we find that is a really nice structure to deliver the content, because then you can have your questions answered in a timely fashion, but it doesn't we don't risk kind of derailing progress of the course. But time permitting, we'll have a little brief Q&A at the end of each section or each module. Yeah, so those are the broad outlines of the class. It's a 4-day course. As you know, today is day 1. We're just kind of getting started today. We are going to first look at the computing environment that we're going to be working in. We're going to be using Microsoft Remote Desktop to access a virtual machine that we're running on a server. And we're going to be in a Linux OS. We're going to be in Ubuntu, running on a remote server. All of you should have received login credentials from Andre. So check your email for those if you haven't already. And the first bit of the course, after these broad introductions, we're gonna walk you through the process of getting on the remote desktop so you can access the virtual machine. And all of the slides, all of the code, all of the materials for the course will be available through the virtual machine. Of course, you have access to the slides through the Dropbox that Statistical Horizons provides, but the slides are also available within the virtual machine, as are 4 R scripts that contain all of the computing exercises that we're going to do over the 4 days of the course. So that's probably helpful code for you to hold on for future use. But yeah, we'll get to that shortly. Kind of getting up and running with the virtual machine. And as a slight aside, the basic structure of the course is, we'll go through some slides, we'll introduce some material, and then we'll get to a computing exercise. I'll briefly describe the computing exercise, and then we're going to go ahead and toggle over to the virtual machine and run the code. So you can do that in tandem. You can do that on your side running the code, or you can just kind of watch me do it. But it's available to you either way. And in that way, we kind of dovetail the substantive information with the actual execution of the methods or the data analysis, which I find to be very effective. So that's essentially the course in a nutshell. So it's 4 days. Day 1, we're going to be introducing the computing environment, statistics, and genetics. Day 2 is kind of like the GWAS day. Primarily, we're gonna look at how do we manipulate and manage big genomic data. How do we do quality control on our genotype data prior to our GWAS? Then we're going to run some GWAS on day 3. We're going to do GWAS post-processing, advanced GWAS methods, and introduce polygenic scoring. And then on day 4, we're going to kind of blast off into the future by looking at really the very cutting edge of polygenic scoring, genome by environment interaction, and genomic SEM. So that's the course in a nutshell. So now let's go ahead and move on to our introduction to genetics. So to kind of set the stage, before we get into the machinery, the molecular machinery of DNA, and sort of how we approach associating genotypes with phenotypes. Let's just kind of set the stage with some general theory about genetics. Maybe one of the most central theories to molecular biology is known as the central dogma of molecular biology. And the figure on the left side of the screen and the figure on the right side of the screen, they basically describe exactly the same thing. Just the one on the right side of the screen does it with more detail. So let's start on the left side of the screen with this figure. And basically what the central dogma of molecular biology tells us is that the flow of information, overwhelmingly, although there are some special cases where these principles do not hold, but in general, the flow of information, of biological information, proceeds from DNA to RNA to protein. So we kind of see this causal structure here, or this causal pattern with DNA creating RNA and RNA creating protein. And then DNA is also self-replicating. So DNA can create itself as well. But basically, and again, we'll discuss some special cases where these principles don't hold. But basically, we now know that that DNA is kind of the blueprint from which RNA is created, which is deoxyribonucleic acid, ribonucleic acid. So DNA is the kind of the origin. It creates messenger RNA, which essentially serves as instructions for creating proteins. That messenger RNA then migrates out of the nucleus to ribosomes, where it serves as instructions to transcribe proteins. So in blue here we see the primary flow of information. Now, in some cases, typically with viral organisms, RNA can be self-replicating, and RNA can in some cases create DNA. But these are very much exceptions to the rule, particularly in kind of higher order eukaryotic organisms. For us, like humans and similar animals, the blue arrows describe the flow of information. So it begins with our DNA. The DNA is transcribed into messenger RNA, which are essentially like kind of recipes. Those recipes migrate out of the cell nucleus into these little organelles called ribosomes, where they instruct the protein on, or they instruct they contain the instructions for synthesizing all the proteins that comprise the bodies and structures and functions of the organism. So on the right side of this slide, we just have it in more detail with some of the enzymes labeled. DNA polymerase is responsible for DNA replication. Over here, the orange arrows depict the flow of information under the central dogma, and the green arrows depict the kind of exceptions to the rule. Again, those exceptions to the rule primarily seen in viruses. So then the transcription. This is the process in which segments of DNA corresponding to genes typically are transcribed into little scripts of RNA, messenger RNA we call them. That's facilitated by an enzyme called RNA polymerase. Then the kind of positive strand of the messenger RNA sort of migrates out of the nucleus of the cell into an organelle called the ribosome. And then at that point, the instructions or the recipe contained in that messenger RNA is then translated into a protein. And then that protein does, all of the myriad functions of an organism's body. And then, we've got the special cases here in the green, less relevant to what we do. This would primarily be relevant to virologists. So this is kind of like a central tenet of molecular biology, that the flow of information is from DNA to RNA to proteins. Okay. Now, in terms of the actual physical structure of DNA, we now know that it is a double helix. We've known that since 1953. On the left side of the screen, we have a figure that kind of depicts it. Yeah, I mean, really, the way I like to think of the physical structure of DNA is it's essentially a spiral staircase in which the rails are sugar phosphate backbone. These are like kind of unimportant. I mean, obviously they're important to give structure to the DNA, but from our perspective, they're less important than the nitrogenous base pairs that kind of comprise the steps of the spiral staircase. So the nitrogenous base pairs that comprise the steps of the spiral staircase, these are the parts that vary in some cases across individuals, and they're what gives rise to genetic variation in humans, right? And you can kind of think of them as a linear sequence of letters, right? There are 4 different nitrogenous bases, which we depict here as A, C, G, and T, adenine, cytosine, guanine, and thymine. And the G's and the C's always pair, and the A's and the T's always pair. And the kind of sugar phosphate railing, they're just there to kind of give structure to the whole thing. But really, from our perspective, we can kind of think of the human genome or any organism's genome as a sequence of letters, the sequence of these 4 letters. And there are about 3.2 billion base pairs in the human genome, but most of them don't vary across individuals. Only about 0.4% of our genomes vary across individuals on average. So the vast majority of the genomes between humans are exactly the same, but there is variation in the sequence of these letters that essentially spell out the human genome. Again, the double helix structure of DNA it's generally attributed to Watson and Crick from their Nature paper in 1953. But in historical retrospect, we know that other folks made meaningful contributions to the discovery of the double helix physical structure of DNA, including Rosalind Franklin, who made several contributions to the discovery of the structure of the DNA. Most notably, she took a famous photo called photo 51, which essentially showed the structure. But she passed away before the Nobel Prize was awarded. The Nobel Prize is not awarded posthumously, and so Watson and Crick basically got all the credit there. But certainly there were other folks involved, including Rosalind Franklin, a guy named Wilkins, so on and so forth. So depending on the organism, there are different numbers of chromosomes. Humans are a diploid organism, meaning for every chromosome we have two of. So, our chromosomes are pairs. Most animals are diploid organisms. So we've got 23 pairs of chromosomes. 22 of those we characterize as autosomes. Autosomes essentially refer to chromosomes that are not sex related or sex linked, and then our 23rd pair of chromosomes depicted over here in the karyotype. This is what determines the sex of the human organism, the biological sex. Here we've got a Y chromosome and XY. The Y chromosome, puny little guy, quite small, with very few genes on it, actually. So this would be a male, an individual whose biological sex was male, at least. We call this picture karyotype. These are metaphase chromosomes that have been microscopically photographed, and kind of the color of the banding has been tweaked to make it sort of more visually interesting. But these are actually photographs of chromosomes in the metaphase of the mitotic cycle. So we've got 23 pairs of chromosomes, 22 autosomes, one sex pair. So for example, with chromosome one of these will come from the mother, and one will come from the father. And that's true for all 23 pairs of chromosomes. You inherit one from each of your parents. These chromosomes are estimated to contain between 20 and 25,000 genes. Which actually is a much lower number than geneticists thought prior to the sequencing of the human genome. It was, because we're such complex organisms and there's so much variation across humans, it was thought that probably there were more genes, but it turns out that just about 20 to 25,000. So, the primary mechanism giving rise to genetic variation across individuals in a species, human individuals, is recombination via inheritance. So let's talk a little bit about how recombination works. Right. So here we've got our father's DNA depicted as two pairs, right? Because our father inherited chromosomes from his mother and father, and likewise, our mother's DNA. She inherited one set of chromosomes from her mother, one set of chromosomes from her father. Now, when our mothers and fathers create gametes, these are sex cells, sperm and eggs. When they create those, a process called recombination happens. And what happens is these two, let's say these are chromosomes 1 from both mother and father. They're so they have two chromosomes for chromosome 1, and those two chromosomes, they get spliced up and recombined. That's what's depicted here in the child's DNA, right? So when the mother is synthesizing, her gametes, her eggs, her molecular machinery is going to chop up the chromosome 1 here and recombine it into a single chromosome, and that process is called recombination. And that's what that's a major source of genetic variation in a population. Some genetic variation is due to mutation, which is kind of spontaneous changes in the DNA. But most of the variation that we observe in a population stems from this process of recombination. Now, when the gametes are synthesized, when the father's sperm and the mother's eggs are synthesized, and their DNA, their chromosomes are kind of chopped up and reassembled during recombination. They're actually the recombination takes place in relatively large chunks of DNA. Right. So it's, instead of, you could imagine that the chromosome could be chopped up into 1 million pieces and recombined, but it's not that many, right? It's much fewer. So actually the blocks that you're going to inherit from, your mother's grandfather or your mother's grandmother are quite large, right? So and that's depicted here, right? We see that we don't see like a smooth gradation between these two colors, which would indicate like very fine grained mixing. We get very coarse mixing during recombination. And that creates a phenomenon called linkage disequilibrium, meaning that variants variants that are spatially proximate to each other, that are close to each other, they tend to hang together across generations. There's this spatial dependence of variants being correlated in genomes due to something called linkage disequilibrium. And linkage disequilibrium it's very interesting. It creates some challenges for the analysis of genomics, but it also creates some opportunities. And so we're going to return to linkage disequilibrium several times during this workshop. First, we're going to discuss it as kind of an impediment or something that needs a nuisance that needs to be addressed when we're calculating polygenic risk scores. Because this linkage disequilibrium phenomenon, it basically the outcome vis-a-vis polygenic scores is that that proximate variants are going to be super highly correlated. So we're going to need to thin out the variants to just to address that spatial correlation before we can calculate our polygenic risk scores. We call that process pruning or clumping. So we're going to encounter LD tomorrow. And then on the final day of the workshop, we're going to encounter LD again in the form of LD score regression, which is a very, very powerful technique, but it leverages linkage disequilibrium to allow us to say things about polygenicity, about heritability, and about genetic correlations. So LD, it from our perspective as analysts of genetic data, it creates some difficulties for us that we have to address in the form of pruning and clumping for polygenic scores, but it also creates some benefits for us in that it kind of underpins some of the population genetics theory that allows us to conduct LD score regression, which we're going to discuss on day 4. LD score regression, very helpful. It gives us an estimator of the heritability of a trait. It allows us to calculate genetic correlations. It allows us to estimate confounding due to population stratification. So and all of that would not be possible without this this kind of feature of genomes where there is spatial dependence, meaning that variants that are close together tend to be very highly correlated due to linkage disequilibrium. And then, as we move through generations, linkage disequilibrium gets, gradually kind of whittled down, right? So, in the first generation, you might have large chunks that you've inherited from, say, your maternal grandmother. But as you move through generations, those large chunks are going to get whittled down more and more as linkage disequilibrium kind of breaks it down. So that's sort of inheritance, genetic inheritance in a nutshell. Another very central concept, which we already touched on with genetics, is how do we get from DNA, which is essentially like 3.2 base pairs or letters. It's like a book comprised of 3.2 billion letters. How do we get from that double helix structure to the phenotypes that we observe in humans, right? How do we get there? Well, through primarily through a two-stage process of transcription. Transcription is depicted, down here, and that is where the DNA, a segment of DNA, is used to synthesize its complement as a piece of messenger RNA. And frequently these transcripts, we call them, these pieces of messenger RNA that transcribe a piece of DNA, they generally correspond to genes. Not always. Sometimes they happen randomly and they have regulatory functions, but usually they correspond to a gene. Now, what defines a gene? That's actually not as simple a question as it might first appear, but generally speaking, a gene is defined as like a spatially contiguous segment of DNA that's responsible for a particular protein product, right? So here we've got gene number 1 here. So gene number 1, it's going to get transcribed into basically a recipe for a protein. Then that piece of that transcript, that piece of messenger RNA, is going to migrate outside of the cell nucleus to a ribosome, and then it's going to be translated into a protein. So every three base pair, contiguous base pair combinations are called codons. And depending on what sort of word those 3 base pairs spell, that's going to correspond to a different amino acid. Remember, there are 20 standard amino acids that humans use to create our proteins. And these codons, they take the DNA instructions, they transcribe them into messenger RNA, and then for every 3 base pairs in the messenger RNA, we have a corresponding amino acid, and then the sequence of these amino acids determine the protein. And there's many proteins in the human body. They have many different functions. And so this is kind of how we go from the blueprint that is encoded in our DNA to create sort of recipes that migrate out of the nucleus into the ribosome, and then are converted into sequences of amino acids that assemble proteins. And those proteins then go on to do all the functions of an organism's life, comprising the tissues, the neurotransmitters, various cell types, and so on. So that's transcription and translation in the nutshell. Now, genetic variation. So, previously I mentioned that, we've got approximately 3.2 billion base pairs in the human genome, but most of that is not polymorphic. Most of that is monomorphic, right? It's the same across everyone. Actually, a very small proportion of our genomes vary across individuals, about 0.4% on average. But that 0.4% variation, that's what gives rise to all of the, or not all of the, phenotypic variability. I mean, certainly environments contribute to that as well. But it contributes to quite a bit of the phenotypic variability that we observe in our species. So, here we're kind of unpacking these concepts. So, here we've got our chromosome, metaphase chromosome here. We're kind of unspooling it here as DNA. And then we're kind of looking in at like the base pairs. So here we got an AT, here we got a TA, GC. So this is the actual, the actual language, if you will, of the human genome. It's kind of a linear sequence of these letters or base pairs. Now, if we zoom in on 2 different individuals here, here we see, at this locus or location in the genome, these folks are the same on all of their base pairs, except for the one in the purple box here. Here we actually have a polymorphic site or a variant. So this would be a locus in the genome where individuals vary in terms of their sequence. And here we got CG in person 1 and AT in person 2. Now, is this relevant? Is this relevant to any phenotypes? Maybe, maybe not, right? So maybe it's of no consequence at all to phenotypes, or maybe it has a critical consequence in phenotypes. So in subsequent slides, we're gonna learn, like, how do we, what distinguishes variants that are highly influential phenotypes versus variants that are essentially irrelevant. We'll get to those features, but at this point, let's just note that the majority of the genome is the same across individuals, but occasionally we do have these sequence variants, and they give rise to a lot of the phenotypic variability that we observe across individuals, but not all of it. There's definitely a role for environments as well. So the 4 different bases: adenine, thymine, cytosine, and guanine. Genetic variation, differences in the sequence of DNA among individuals, and a mutation is a newly arisen variant, right? So mutations are the ultimate source of genetic variability across individuals. But sort of a more proximate source of variation is recombination, as we just described. Yeah, that's kind of an overview of genetic variation. Now, there frequently we use this kind of typology or classification scheme where we break genetic variants. And again, these are places in the human genome where individuals vary to one distinct or to one extent or another. We frequently use a typology to kind of break those variants into 2 categories: common variation and rare variation. So a rare variant is typically defined as a genetic variant that is present in less than 1% of the alleles at a given locus in the population. So these would be variants that if we took a large sample of people, that the minor allele, meaning the less common variant observed, occurs in less than 1 in 100 people, right? So so rare variants, they're quite interesting, and they're often quite influential on phenotypes, but they're challenging to study. They're challenging to study because they occur infrequently. There's no guarantee that that a given rare variant is going to appear in your data set, and they also are tricky because of with their frequency being so low, with their occurrence so low frequency, that has a negative impact on statistical power. So if you're dealing with rare variants, oftentimes you're going to need very large sample sizes in order to have the statistical power to reliably detect the real effects. So rare variants, they're important. I certainly wouldn't minimize their importance, but they can be challenging to study because you need very large sample sizes. Oftentimes like biobank sized samples. Common variants, on the other hand, these are genetic variants that are present in greater than or equal to 1% of the alleles at a given locus in a population. So this is the stuff that typically you're going to analyze in GWAS and related methods. You're typically going to analyze common variants over there. And this 1%, there's nothing, it's kind of an arbitrary threshold that's been applied. I didn't come up with it, but the kind of scientific establishment, decide. I mean, 1 is as good a number as any. But it's essentially arbitrary. Sometimes you hear common variants described as 2% or something, but usually 1% is considered the cutoff. Okay, so let's kind of think about rare variants and common variants in terms of their impact on phenotypes, right? And this plot does a pretty good job of that. We've got the effect size. This would be how large an effect the variant has on the phenotype. We've got that on the y-axis. Then on the x-axis, we have allele frequency. This would be minor allele frequency. This would be how commonly the rarer of the two variants occur in the population, right? Rare variants, obviously they, by definition, they have low allele frequencies, low minor allele frequencies, meaning the rarer of the alleles are, rarely appear in the population. So the allele frequency of rare variants is quite low, but oftentimes their effect sizes can be quite large. They can be, rare variants can have big phenotypic effects, which we describe as high penetrance here. And a lot of Mendelian traits, kind of medical syndromes, Rett syndrome or, Angelman syndrome, all these medical syndromes that you may have heard of. They're frequently, those syndromes are often Mendelian, meaning they're caused by one or very few variants. Those traits are generally caused by rare variants. And it's due to selection, they basically have to be rare variants, right? Because typically those individuals don't reproduce. And so selection kind of relegates these syndromes or Mendelian traits, deleterious Mendelian traits. They typically are rare variants. And then kind of moving on the diagonal here, we've got low frequency variants. These are sometimes characterized as uncommon variants, so they're not really rare. They're not super rare, but they're also not common, so they're kind of intermediate. Sometimes they're called uncommon variants, and they typically have intermediate effects. And then you've got common variants. These are the variants where the minor allele frequency is tends to be relatively high. And these are we characterize as low penetrance traits, meaning they don't have large effects on the phenotypes, but that doesn't mean that they don't have any effects. They often do cumulatively. And common variants are often responsible or play a big role in determining the variation in complex traits, right? So we often contrast Mendelian and complex traits. Mendelian traits are traits. These are often deleterious syndromes, and they're frequently caused by one or two rare variants or just a handful of rare variants. On the other hand, complex traits, these are things like height or personality traits or cognitive traits or things of that nature, body composition. And these are what we often refer to as massively polygenic traits. These are traits that are influenced by tons of different genetic loci. There's a lot of debate for a long time, and there's certainly folks in some quarters of genetics even now who argue that most of the phenotypic variation that we observe in humans is not due to common variation; it's due to rare variation. But increasingly, the evidence for massive polygenicity in complex traits, by which I mean the evidence supporting the fact that complex traits are determined by thousands of genetic loci, or if not tens of thousands. That evidence is becoming increasingly overwhelming. I'm going to show some of that evidence, but yeah, so this model, that it's consistent with the evidence that we have. Then off the diagonal here, you've got variants that have very small effects and very low allele frequencies. These are basically impossible to detect, right? Because you just don't have the statistical power to detect it, right? And how important is it really at the end of the day if the effects are very small? Once we have like truly enormous biobanks, in the millions, maybe we can do more with this stuff. But for now, we don't really observe these rare variants with small effects. And then on the other side of the off diagonal, you would have variants that are have a high allele frequency and a high effect size. These are mostly non-existent due to selection, right? Because if you have a trait that's really really increases fitness, it's very quickly going to become universal in the population, or and if you have a allele that has a super highly deleterious effect on the organism, it's going to be kind of removed from the population by negative selection. So there's some cases where there are common variants that have large effect sizes, but it's definitely the exception to the rule. Like ApoE, is a variant that affects like the metabolism of cholesterol. It has big effects on all-cause mortality and cardiovascular traits. It's one of those rare exceptions where it's got a kind of a big effect size, and its allele frequency is often, in most populations, not super. Its minor allele frequency is not super small, but that would be an exception to the rule. Generally, we don't observe common variants that have large effect sizes. And then over here, just a little bit of additional detail from DBSNP too. This is like a database that catalogs all of the variants that we know of. And so to date, about 500 million variants have been identified. About 3.3% of those are common variants. About 3.8 of those are rare variants. Here we're defining rare variants as minor allele frequency between 1% and 0.1%. And then most of the variants that exist in human populations are very rare or novel. And these are occur very rarely. These are very rare, occur very rarely. So mostly we're going to be dealing in this domain, common variants. And if you're dealing with like UK Biobank or a very large sample size, you can also include some rare variants in your analyses as well. But this very, very rare, and sometimes these novel variants are called private variants, meaning that they might only occur in one family in the world. These are they can have very large effects, but they occur so frequently that for all practical purposes they're quite difficult to study using the methods that we're going to discuss. Okay, the range of genetic variation. So another way that genetic variation is another kind of typology that we use to characterize and define genetic variation is sequence variation versus structural variation. So sequence variation, this is variation at the single nucleotide level. So we've got these nitrogenous base pairs, right? These essentially are the letters that comprise the language of the genome. Each of those nitrogenous base pairs we would call a nucleotide. And if we're talking about a single nucleotide, like a single A, C, T, or G, then we would call that variation at that level a sequence variation. And if you've got a substitution at a single nucleotide, meaning at a certain locus, a single nucleotide locus in the genome, you've got two alleles. It can either be, say, an A or a C. That we would call a single nucleotide variant. And if it's a common single nucleotide variant, then we would call it a SNP, or a single nucleotide polymorphism. And so SNPs are primarily what we deal with in kind of statistical genetics. But there are other forms of sequence variation as well. There's also indels. Indel is like a combination of the word insertion and deletion. And these are places where you might have one base or a few bases that occur in some individuals in the population, but are absent in other individuals in the population. So those are called indels. So yeah, and SNPs and indels, or single nucleotide polymorphisms or variants, and indels, These are essentially, categorized as sequence variation. They're considered sequence variation because they occur at a single locus. They're variants that occur at a single locus, and that's where most of our common variation comes from. Our common variation is almost entirely sequence variation. But in addition to that, we also have structural variation. So structural variation is the sort of statistical genetics is starting to study structural variation to some extent, but structural variation has historically been the purview of medical geneticists because these are typically rare variants. They typically are highly penetrant, and they often have huge phenotypic, generally deleterious phenotypic consequences. So our syndromes, like earlier, I was Rett syndrome, Angelman syndrome, Down syndrome. Those are all caused by structural variants. Structural variants, again, typically rare and highly penetrant, meaning they have big effects on the phenotype. And oftentimes they're responsible for Mendelian disorders. And again, disorders that are caused by, a single or, a handful of genetic variation. And we can think of structural variants, we can kind of categorize them by how large they are, right? So you can have kind of small structural variants like variable nucleotide tandem repeats, microsatellites, mini satellites. You can have larger indels. We discussed indels over here with sequence variation, but if you've got a large indel, which maybe it's 100 base pairs, then that wouldn't be considered sequence variation per se. It would be more of a structural variant. You can have inversions. Inversions are where a chunk of DNA it basically gets flipped, and so it reads backwards, which, depending on where that occurs in the genome, can cause problems, right? It can cause different problems with protein manufacturing. Di, tri, tetranucleotide repeats. These are rare. They're just kind of unusual nucleotide variants. Then kind of moving to a larger resolution, 1 kb. This is 1 kilobase or 1,000 base pairs to submicroscopic. Here we got copy number variations. Copy number variations are increasingly recognized as being quite important to phenotypic variability. This is like, if you have a chunk of DNA, maybe some people have one copy of that chunk. Some people have 2 copies of that chunk. Some people have 10 copies of that chunk. And that can have big phenotypic consequences. So that's copy number variants. Segmental duplications. This is when you've got a piece of a chromosome that's been duplicated. Inversions, we talked about that up here, but you can have larger inversions. You can also have translocations. This is when maybe a chunk of, let's say, chromosome 8 breaks off and grafts onto chromosome 22, right? That's when chunks of DNA swap to different positions in the genome, oftentimes different chromosomes, and that can cause a lot of problems. A lot of leukemias are caused by genetic translocations in the bone marrow. Micro duplications, micro deletions. These are kind of like larger scale insertion and deletions. And then, I'm not going to go through every bullet point here in the structural variation, but some structural variations are quite large, right? So Down syndrome is typically caused by a trisomy, which means for a given chromosome, I forget which chromosome it is, but instead of just 2 pairs, two chromosomes, you actually have a third chromosome. So that would be considered a structural variation as well. And then marker chromosomes, these are just kind of like little extra chromosomes that you might have. Ring chromosomes, isochromosomes, these are sort of weird structural transformations that can happen to chromosomes, typically very deleterious to the fitness of the organism. But so I guess if we zoom back a little bit, the take home here is that that we've got sequence variation. This is most of our common variation, and this gives rise to a lot of the variation that we observe in complex traits, which is the primary focus of this class. And then we've got structural variation. Structural variation typically rarer. Lower minor allele frequencies, and oftentimes highly penetrant, meaning it has a big effect on the phenotype, and typically deleterious, meaning it's bad for the health or the fitness of the organism. And so structural variation is largely the purview of medical geneticists. Sequence variation is of wide interest to anyone with interest in complex traits. But one particular, so the last thing I'll say about the slide, and then we need to move on. One particular type of structural variation, in particular copy number variance, this we're finding out is often quite important for complex traits as well, copy number variance. And sometimes CNVs, which is the acronym for copy number variance, sometimes those interact with sequence variance. In kind of epistatic ways or kind of genetic interactions. So all of that is to say, most of what we're going to learn in this course is going to be focused on sequence variation, but copy number variants are an increasingly kind of exciting area in complex trait genetics as well. It turns out that it seems like they have pretty big effects on complex traits. Okay, so here's just kind of trying to reinforce our understanding of SNPs, single nucleotide polymorphisms. This is a variation at a single base pair. Sometimes it's referred to as a nucleotide substitution, but basically, so at every locus in the genome, we have two copies, one that we inherited, on the one chromosome from our mom, one that we inherited from a chromosome from our dad. And so this first individual at this SNP has the TT genotype. So they have the thymine in both chromosomes. The second person here has a TC genotype, meaning they inherited a T from one parent, a C from another parent, and then this third person has the CC genotype here. So the fact that this is not uniform across individuals, that it varies across individuals, is what makes it polymorphic, right? And at some point in the historical lineage of this organism or this population, a mutation arose. So maybe C was the ancestral allele, and then at some point someone had a mutation, and suddenly they have a T there. Turned out it wasn't so bad for their fitness, so it kind of made its way into the population. That's kind of how these variants make their way into the population. They occur as a mutation. If that mutation has a fitness benefit, then it generally will increase in frequency in the population across subsequent generations. If it's neutral to fitness, meaning it doesn't make a big difference on the success of the organism, it might be maintained at a certain level, or depending on genetic drift, maybe becomes more or less popular. But if it's bad for fitness, if you have a mutation that's bad for fitness, typically those get eliminated from the population relatively quickly through negative selection. Like the folks who have it are less fit, and so the declines over time. Okay. So generally, each SNP can take two different alleles. So in this case, it can either take a T or a C. And then, after a SNP has been identified and it's been validated, eventually it gets an RS identifier. And this is like a unique identifier that every SNP has. And you can bang these through DBSNP or various online databases to get information about any given SNP. Okay, and then here we have a slide describing indels. Remember, indels is our second type of sequence variation, and these are at a given loci. You either have the insertion or you do not. or you could look at it from the other side. Either you have the reference allele or you have a deletion of it. So this individual has one copy of the, say, the reference genotype here, which is GCT, and then they have one deletion. They're missing that genotype from their parental chromosome there. So we would say one reference, one deletion for this individual. This individual has two reference, meaning that they have the indel on both chromosomes, and this individual, the third individual, we would characterize as deletion. They do not have, they did not inherit that indel from either of their parents. They did not inherit that the reference genotype from either of their parents. So we would characterize them as a deletion. Okay. So again, SNPs, indels, these are our sequence variants, and these are primarily what we analyze when we're doing complex trait genetics. Okay. Structural variation. We already talked a good bit about structural variation, so I'm not going to spend a ton of time here. But there are various primary types of structural variation. We've got duplication. So this would be a situation where a segment of DNA gets replicated in a mutation. So as opposed to having the reference allele, which would just be kind of one copy of this locus, now you've got two copies of it. That's a duplication. Deletion's kind of the reverse of that, right? It's a segment of DNA that gets lost along the way in some in some individuals. An inversion. Remember, an inversion is when a chunk of DNA gets flipped and spliced back into the same locus, or gets flipped into the same locus, which in some cases is harmless, right? But if one of the boundaries of that flip is within a gene or an important regulatory region, it can cause some problems. We got an insertion. That's when a novel chunk of DNA gets kind of spliced in. And then we've got copy number variation. So this first individual here, we would say they have a 0 copy number variation at that locus. This second person has a 1 copy number for this CNV. This person has a 2 copy number for the CNV. And again, CNVs, this is kind of an exciting area of research in complex trait genetics. We're now realizing that these CNVs are they occur frequently, and they often have large phenotypic consequences to complex traits. Okay, so far we've kind of talked about the physical structure or sequence of DNA, but we can also look at it from a functional perspective, right? So what does the DNA do? And it turns out that there's a lot of variability in what DNA does, right? So previously we discussed this concept of a gene, right, which is a contiguous segment of DNA that's generally defined by its ability to produce a protein, a given protein. But it turns out that the vast majority of our DNA is not in genes; it's between genes. We call it intergenic DNA. That DNA used to be called junk DNA. Because it wasn't, initially geneticists thought that it was just kind of like a lattice to kind of keep the genes together, like, kind of duct tape to keep the genes together. But over time, we've realized that that intergenic DNA, it can actually be super important. It can regulate the expression of genes, and we'll talk about that in the next 2 slides. But this slide focuses on genic DNA. This is DNA that occurs within genes. And this is also, this is the DNA that has the most obvious functional consequences to protein production, right? And that's because, it's the DNA in genes that encodes the proteins, right? Because remember, genes are the unit, the kind of contiguous segments of DNA that are responsible for coding a given protein. Remember, they get transcribed into RNA, and then those RNA instructions are used to create the proteins. So if we have variants within genes, those can often have large effects on proteins. They can, they can change what proteins produced, or in some cases, they can cause the protein to be non-viable, which is often quite bad. But so let's begin discussing genic DNA by looking at this figure in the upper right hand part of the slide. So let's consider this as a gene, and then each gene can be described as a combination of exons and introns. And over here, there's something called a promoter that we'll discuss briefly. But basically, exons, these are the parts of the DNA that are actually used to transcribe the RNA that's then going to make the protein. The introns, these actually get snipped out of the RNA transcript before the protein is made. So in some ways they're less important because they don't have direct consequences on protein production, but they often have regulatory functions. So oftentimes they get snipped out and they serve as like these kind of small small RNA molecules that float around and have regulatory functions. But the exons, this is what actually gets transcribed into RNA and translated into proteins. So the exons, they are quite important. If you had to choose a feature of DNA to focus on, you could do much worse than focusing on exons, because the prior probability of them being involved in phenotypic variation is much higher than other areas of the genome, because they directly translate into proteins. Okay, so here we got a gene. So this is genic DNA, and we can kind of parse genic DNA into the exons and the introns. So the introns kind of get snipped out at the RNA stage, and then the exons get knitted together into the transcript, and then that's what gets translated into the protein. Now, we can also break down the types of variants further using this kind of probability tree. So, a genetic variant can be in a non-coding region. So these would be our intergenic SNPs. These would be SNPs that don't appear in genes. So we would say these are non-coding because they don't directly translate into a protein. But again, we wouldn't necessarily say that they're unimportant, right? Because they might have regulatory functions. So even though they don't contribute to the translation of a particular protein, they can still have big effects on how much of a protein is made. So they can have big effects on the gene expression rates. So non-coding regions, they used to be thought of as junk DNA unimportant, but now we know that junk is probably not the best descriptor for them. They can have gene expression effects. Now, if they occur in a gene, and if a variant occurs in a gene and it occurs in the exon of a gene, we call it a coding variant. And those coding variants can be broken down further into synonymous and non-synonymous coding variants. A synonymous coding variant does not result in a change in an amino acid. So synonymous variants usually, don't have big effects on the protein products or the eventual, kind of downstream phenotypic results. I mean, they can in some cases, but they don't change the amino acid sequence, so they don't have like dramatic effects on the proteins. Non-synonymous variants, these are the ones that are often very interesting. They cause a change in the amino acid sequence, and that change can either be a missense change in which one amino acid turns into another one. So, obviously that can have a big effect on the protein. It can create a different protein product. In fact, it does. But, maybe that protein is still viable for its purpose. Maybe it isn't, right? But missense non-synonymous variants, these are variants that occur in exons, and they result in the change in an amino acid in a protein. So these potentially can be quite important. But nonsense, non-synonymous, these are kind of the worst of all worlds. So these basically cause a premature stop codon, which tells the protein to stop at that point prematurely. So oftentimes these result in non-viable proteins. So these are often like pretty harmful variants. So this is kind of an entry point to thinking about functional variation in the genome. So we can talk about structural or sequence features of the genome, and these describe like the physical apparatus. But then there's also differences in the function of these variants, right? So on one hand, we've got intergenic variants. These do not directly affect protein manufacture. But they can affect the rate at which genes are transcribed, the rate at which proteins are produced. And then, within the exons of our genes, then we have coding variants that can be broken down further in this way. So that's a little overview of functional variation. Now, so here we, just a little bit more elaboration on functional variation. On the left side of the slide here, we have our coding SNPs. Remember, these are the SNPs that occur in exons. And here, we've got two different non-synonymous SNPs illustrated. This one would be a missense, where it does not result in the change of amino acid, or it does result in the change of amino acid, but maybe the protein is still viable. Here, this would be a non-synonymous nonsense SNP, and that causes a premature stop codon, basically, generally causing the protein to be non-viable. So these would be coding variations. Now let's talk about non-coding. And this is more indirect, but also quite interesting, right? So these are genetic variants that do not take place in genes, and sometimes they take place quite far from the genes that they moderate the expression of, the genes that they influence. And these intergenic SNPs, which can have effects. They can have effects if they occur in certain regions of the genome, which we call enhancers or promoters or repressors. And these are non-genic loci within the genome. But the way that they work is, let's say we've got a SNP in this enhancer over here. It recruits these proteins called transcription factors, and then those transcription factors in turn attract a complex called the activator complex, or co-activators. And when that bonds to the promoter of the gene, it can affect the expression of the gene. It can silence the gene, preventing it from producing any protein product, or it can upregulate the production of the gene, resulting in more of a protein product being expressed. So these variants that occur in non-coding regions, if they occur in enhancers, promoters, or repressors, they can actually have big effects on gene expression, even though they don't affect what protein is produced by that gene. And then over here on the right side of this slide, I just have like a slightly more elaborate diagram describing the transcriptional machinery of gene expression, right? So here we've got the promoter. The promoter is always immediately upstream of the gene, and enhancers and conversely repressors, even though they can be quite far away, in the linear sequence of the gene, in the linear sequence of the genome, they can be quite distal from the gene that they affect. But once they start recruiting transcription factors, they can kind of spatially bend to physically interact. So the DNA loops around and then spatially interacts with the promoter. And then, if enough transcription factors create a critical mass that attracts the activator complex, then it initiates transcription of that gene. So you can see that genetic variants that occur in enhancers or repressors or promoters, they can have big effects on gene expression, even though they're not directly involved with protein production. They're actually quite important and an area of a lot of focus currently in research.